Hello and welcome to our last lab of the term. In this lab we will discuss different blood groups and learn to determine those groups experimentally. Blood types are determined by the special cellular markers present on the surface of the red blood cells. In fact, every cell in your body carries special identifiers on the surface of the cell membrane. They're called antigens. Your immune system is trained to recognize your antigens as self and does not generate any kind of immune response against your own cells. So red blood cells, like any other cells, carry antigens unique to them. In many cases, plasma may contain antibodies against the antigens foreign to your blood. Also, we know over 30 different types of blood groups. There are only few that have clinical importance. ABO blood types and Rh blood types cause a severe transfusion reaction, as well as Rh blood groups may cause problems with pregnancy. That's why we will study them more than others. Other blood groups like MN blood groups, Duffy, Lewis, and so on, usually used in legal disputes, in fraternity tests, as well as in criminalistics. Let's talk a little bit about the ABO blood groups first. In general, humans generate two different types of antigens of this particular type: A antigen or agglutinogen and B antigen. So individuals may have none of them, this is blood type O, may only have A antigen, this is blood type A, may only have B antigen, this is blood type B, and may have both A and B, and this is blood type AB. Simple enough? So again, blood type O, people do not have antigens or agglutinogens of that particular type. In blood group A, they only have A agglutinogens. Blood type B, they have B agglutinogens. In AB, they have both A and B agglutinogens. Okay? All right. So let's practice a few questions. Determine the blood group if there are no agglutinogens of ABO type present on the red blood cells. Blood type O. Determine blood group if there are A agglutinogens present. That's simple. Group A. When we are born, our plasma does not have any antibodies of the ABO type. But as we continue our living, we get exposed to different microorganisms. For example, E. coli populates our gastrointestinal tract, and some of those microorganisms get in our bloodstream, and of course our immune system efficiently deals with them and develops antibodies. But some of the antigens on the surface of the E. coli bacterium mimic antigen A, and therefore we will develop anti-A antibodies. Now, of course, if you have blood type A, you will not develop that particular antibodies. You will develop different type of antibodies to fight E. coli, because again, that's not the only cellular marker. Streptococcus that populates profusely our skin may mimic antigen B. Even small cracks on the skin, some of that microorganism gets in our bloodstream, and we develop antibodies against that antigen. So we develop anti-B antibodies. Okay? If you have a blood type O, and of course you will be exposed to E. coli and Streptococcus, you will develop anti-A and anti-B antibodies. Make sense? If you have blood type A, you will develop anti-B antibodies only. You will not develop anti-A antibodies because that particular antigen will be viewed as self. If you have blood type B, anti-A antibodies will be developed, but no anti-B antibodies. Blood type AB will not develop any antibodies of that particular type. To put it all together, if you have a blood type O, your red blood cells have no agglutinogens or antigens, but the plasma contains antibodies or agglutinins, anti-A and anti-B type. If you have blood type A, your red blood cells are equipped with antigens or agglutinogens of type A, and plasma contains anti-B agglutinins. Blood type B contains red blood cells equipped with B agglutinogens or antigens, and anti-A agglutinins or antibodies are found in plasma. Finally, in blood type AB, red blood cells contain antigens of both A and B type, and plasma does not contain antibodies of these types.
There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Let's practice couple questions. Determine the blood group if there are only anti-A agglutinins present. Only anti-A, that's blood group B. So the answer, blood group B. Determine the blood group if there are no agglutinins of ABO type present. No agglutinins present, that's blood group AB. Okay, very good. Determine the blood group if there are anti-B agglutinins present in plasma. Well, you can answer that already. So that would be blood group A. To confirm, A agglutinogens present on red blood cells. So here you have 2 pieces of information to confirm your answer, blood group A. The difficulty here is predominantly in terminology. Agglutinogens are antigens, agglutinins are antibodies, and all of the words start with A. So if you get your terminology straight, you should be just fine determining blood groups.